Hello, Indiana Virtual. The purpose of this video is to give you an introduction to your next lesson, 0304, Hereditary Patterns. So please go ahead and open up the lesson. And like always, scroll down to the bottom of that first lesson to find the biology notebook. Feel free to open it here or go to our Google site and open the notes handout here. Okay, let's give a little introduction to this lesson. In this lesson, you'll learn the principles of probability. You will complete various interactives using Punnett squares for both monohybrid and dihybrid cross practice. Ultimately, you will relate the influence of genes on the development of traits. Patterns of inheritance will be demonstrated throughout this lesson. After working through the content, you will submit responses to 10 multiple choice questions for grading. So you got a multiple choice assignment on this one. The multiple choice assignment is worth 20 points. Okay, let's open up the notes handout and let's look to see what we're going to learn in this lesson. Okay, so we're going to start learning about probability. So we are going to involve a little bit of math in this lesson. Nothing too crazy though, but that's what probability is, the chance that something will happen. And in this case, the chance that you will see a certain phenotype or a genotype when you crossbreed two parents together. Okay, we have some vocabulary that we're going to look at here. We have homozygous, heterozygous, genotype, and phenotype. Okay, and then we're going to compare those two words, genotype and phenotype. You're going to learn about a manipulative, a square, a box that's divided into 4 squares called a Punnett square, named after the scientist that developed this little box, I guess. And we use it to determine probability with monohybrid and dihybrid crosses. Okay, and then we will learn about something called a test cross. And then we will apply that information, both the Punnett square information, test cross information, your terminology, genotype, phenotype, homozygous, heterozygous, in practice called a monohybrid practice. Monohybrid, that prefix mono means one, so we are looking at one trait at a time. For example, this first scenario that we have, we are looking at the height of a plant. Height is the one trait, and we are looking at our plant either being tall or short. Okay, but it's considered a monohybrid one trait because we are just looking at height, although height has 2 different features, tall or short in this case. Okay, then we're going to go on to a topic called independent assortment. And then we will have dihybrid practice. The prefix di means 2, so we're looking at 2 traits at a time. So maybe for the pea plants, we're looking at height, and the plant could be tall or short. And maybe we're looking at color. The plant could be purple or white. Okay, so we're looking at 2 features, 2 phenotypes, and looking at the probability of inheriting any combination of those 2 phenotypes together. Then we have a section called extended genetics and sex-linked traits, and then something called a pedigree. A pedigree is a genetic family tree. Okay, so you have a couple of sample questions here that you'll be able to work through and practice with as well. Okay, with that, and remember, take very detailed notes. Jot down as many examples as you can. Really include some super detailed information and vocabulary so you can use this notes handout on your upcoming quiz for this, the 10 multiple choice questions. All right, you see we have 7 slides, so you will read through this first slide, the introductory page. When you're done with that and documenting anything that you need to on your notes handout, then go on to slide 2. Here we're looking at hereditary patterns, and you're learning about probability, the chance that something will happen. They have, excuse me, they have an example of flipping a coin. What's the probability if you flip a coin, the coin will show up with heads, or what's the probability that tails will be visible? Okay, so you'll work through the concept of probability, and then we're going to start applying that to genetics and learning first about some vocabulary, homozygous and heterozygous, and then genotype and phenotype. Okay, so we've got another diagram here that compares, really compares that terminology. What is a genotype? What is a phenotype in comparison to one another? What's the term homozygous mean versus heterozygous? Okay, So once you're done with that slide and documenting that information, go on to slide 3. We then have Punnett squares. You have some instructions here on how you work through a Punnett square, how you set up a Punnett square. Okay, so make sure you read. There's step 1, and we have A, B, C, D here. So you really want to go through this in detail, and you want to make sure that, use some extra sheets of paper. Work on this by hand. Punnett squares are like math problems. You just need to keep doing them until you get enough practice and you feel good about being able to solve a Punnett square problem. Okay, once you got it, you got it. But you have to have the terminology down first, genotype, phenotype, homozygous, and heterozygous, before you're going to be able to be successful on Punnett squares. So once you have that terminology down, just practice Punnett square after Punnett square until you got it. And once you got it, then you're good to go. Okay, so they're going to work through how to solve, or how to set up a Punnett square, and then how you solve for that Punnett square, and then how you interpret your information in the end. Interpret your conclusions, determine the results in the end. Okay, so once you are through that, you're going to learn a little bit about something called a test cross. So what a test cross helps us figure out is if we don't know what one of our parents' genes are, we can use the offspring to work backwards to figure out our parent. Okay, then you've got some practice questions here on monohybrids. Okay, when you're done with that, continue on to the next slide, slide 4, and you'll learn about the concept of independent assortment. You have some interactives here that you'll go through and document data and information on each of those. And then the lesson rolls into dihybrid practice. Okay, once you feel you have that down, go to slide 5, where you will be learning about extended genetics, some exceptions to the rule, some things that Gregor Mendel didn't know. Gregor Mendel laid the foundation for so much of genetics, all of the basics: dominance, recessive, the parents, F1, F2s, probability, how probability applied to genetics. So he really laid the foundation for us. There are lots of topics, though, that he didn't know existed when he was doing his research. So you're going to learn about 4 exceptions to Gregor Mendel's rules. So we have incomplete dominance, we have codominance, we have multiple alleles, and polygenic traits. So you want to make sure that you document that information as well. Definitions and examples of each would be great. And then go on to sex-linked traits. So some of our traits are found on our X or Y chromosome. We all have 23 pairs of chromosomes. 1 through 22 are considered our autosomes, our non-sex chromosomes, while chromosome number 23 are our sex chromosomes. And if a person is female, that person has 2 X's. If a person is male, they've inherited an X and a Y. And so there are certain features that we inherit that are attached to those chromosomes, that are coded for by those chromosomes. So any trait that's connected to the X or Y chromosome is considered a sex-linked trait. So we can also have some disorders that are sex-linked disorders, that they're only attached to that X or Y chromosome. And sometimes they can be more common for men to inherit certain diseases if they are on the X chromosome, because men only have 1 X chromosome. So sometimes it's easier for a male to express that trait versus a female who has 2 copies. And Y-linked traits, only males can express those, as females do not even have the Y chromosome. Okay, so once you have finished learning on that slide, go on to the next. Now you'll learn a little bit about a pedigree. I love pedigrees, genetic family trees. They're fun to analyze. I actually just made a pedigree for my family for a class here at the beginning of this school year. So you'll learn about a pedigree and what the symbols and shapes mean. And you have a couple of questions here that you can check your answers as well. And once you're done with that, then go on to the final summary slide. And once you have documented, you think everything you need to know, and you have written every possible note that you think you could have that's important on your notes page, then go on to the assignment, the 10 multiple choice questions. Click on start. And many of you have. If you have questions on these, feel free to send me an email. And all I ask is that when you send me the email, when you say you're confused on a question, I need to see the question. I need you to copy paste the question and the answer options to me. And then I need to see your thinking. With each answer option, I need you to tell me why you think that one is an answer or why that may not be an answer. So that way we can have some dialogue and discussion back and forth to help you work through that. All right, guys? So I hope you enjoyed this lesson. This is one of my favorites. So good luck.